In this lecture, we're going to continue with the evolution of photosynthesis. We will have a short recap of what we learned about cyanobacteria and then take a look at how algae and how land plants evolved. From this, together we will have a look at the emergence of a new photosynthetic module, the light harvesting complexes, and also talk about some specific light harvesting complexes and how they are important in photoprotection. So let's take a recap at cyanobacteria. Cyanobacteria are the only photosynthetic prokaryotes that perform oxygenic photosynthesis. They incorporate both types of reaction center modules in one electron transport chain, and they also have a specific light harvesting antenna called the phycobilisomes. These cyanobacteria are quite important as they are involved in the development of photosynthetic eukaryotes, which formed very likely from an endosymbiotic event, and they are an ancient eukaryotic cell engulfed an ancient cyanobacteria and formed the first algae. These algae developed further into lineages that we still know today, namely the glaucophytes and the red algae and green algae. This primary endosymbiosis event is very important as it gave rise to the first chloroplasts that are still present today in modern plants and algae. We're not going to talk about the specifics of how this evolution took place. Instead, we're going to focus on the further development of the algae. So red algae and green algae development incorporated more endosymbiotic events. I'm not going to talk the details in there because there are quite many and not so important for understanding the bigger picture. So I will just focus on a few. The red algae secondary endosymbiosis gave rise to what we now know as cryptophytes, which later developed on further into haptophytes, stromatophiles, which incorporate also brown algae, and diatoms, and the alveolates, which incorporate the dinoflagellates. In the green lineage, there was also a secondary endosymbiosis event, which gave rise to the chlororanchinophytes. In the further development of the green algae, that didn't incorporate further secondary endosymbiosis or tertiary endosymbiosis, but they gave rise to what we now call the green lineage to land plants, which involved streptophytes, the first development of land plants such as hornworts, liverworts, and mosses, and the vascular plants. We will have a short look on this green lineage of land plants as it evolved, but only a very short one. So, as I mentioned already, chlorophytes developed into streptophytes, which were probably the first algae who colonized the land. From these developed the liverworts, mosses, and hornworts, which we still call like very basal plant species. Further in the development, it got developed some vascular tissue, which gave rise to the lycophytes and the true ferns, or monoliophytes. From that, the seed plants emerged, what we now call the gymnosperms, and also the angiosperms. The angiosperms are also called flower plants. For simplicity of this talk, I will refer to all higher plants as vascular plants, and specifically when we go into details of the LhCs. So for now, this is the basic overview of the evolution of algae to land plants that we need. So now let's switch to the last part of this talk, the photosynthetic light harvesting complexes. As I mentioned already, it's a new antenna system, different from the phycobilisomes, what we know in cyanobacteria. They are embedded in the membrane domain, so they have a specific free transmembrane helices, as you can see here, that are all conserved in all LhC types. Also, they bind different pigment molecules, so called chlorophylls and carotenoids, that make them distinguishable from each other. So now let's have a look at how they are distributed in the algae and land plants. Glaucophytes never joined the winning team of algae. They always kept their phycobilisomes. Different are red algae, the rhodophytes, which developed already the LhCs, but mostly for photosystem 1, but kept phycobilisomes in their photosystem 2s. In the further development of red algae, the younger types, the cryptophytes and haptophytes, cryptophytes lost part of their phycobilisomes, only having residual luminal phycobilisomes, but already had LhCs for the rest, and this continued in the haptophytes and spermatophytes up to the alveolates. In alveolates, there's also a special kind of LhC, which has a very different or very special carotenoid chlorophyll protein called the PCP, which is again, however, beyond the scope of this topic. In the green lineage, LhCs got dominant quite quickly and immediately took over and served as antenna of both photosystems and did not change in the evolution of land plants. To complete this picture, now let's have a look at how the chlorophyll and carotenoid composition of each in these different groups are. So in the rhodophytes, you only find chlorophyll A, and there's a major carotenoid, zeaxanthin. This is already different from the younger evolutionary red algae groups. So in cryptophytes, you have, for example, only chlorophyll A or chlorophyll C bound to the LhCs, and alloxanthin as a carotenoid. While in the different groups, you find fuchsoxanthin, or in the special type that I mentioned already, also the pyridine. This is very different from the ones in the green lineage, where the LhCs mostly bind chlorophyll A and chlorophyll B and have as their major major carotenoid, lutein. These differences between the various carotenoids and chlorophylls lay in the small molecular modifications which slightly change the absorption spectra of these molecules. This on the one hand gives them different colors to our eyes, but is also important for photosynthesis, as this allows them to use slightly different colors of light for photosynthesis and gives them certain advantages, for example in the water column, where not every light color is penetrating to the same depth. In addition to the normal LhCs, which main function is in light harvesting and transferring the energy to the photosystems, there are also kind of special LhCs, LhC proteins, which function mainly in photoprotection. This photoprotection is needed when the amount of light availability exceeds the photosynthetic and metabolic capacity. We call these excess light conditions. In simplified terms, the photoprotection is achieved by two different mechanisms, which are still not fully understood. The one is the direct quenching of excess light via pigments, and the second one is induction of structural changes in other LhCs triggered by a decrease, for example, in the luminal pH. These specialized photoproteins that I mentioned are the LhcSRs, or also called in some species LhcX, and the PSBS protein. It is, however, important to note that all LhCs have some degree of capacity for photoprotection. It's just that these ones are specifically evolved in that direction. One of them is LhcSR or in the LhcX group, which is involved in direct quenching and induction of structural changes, while the closely related PSBS protein is not directly related to direct quenching because it doesn't bind any chlorophylls or carotenoids, but very likely induces structural changes and therefore lets other LhCs serve as a quencher. Now let's go back to the phylogenetic tree and have a look at how they are distributed in the different algae and land plants. If we take a look at how the LhcSRs and LhcX proteins are distributed in algae and plants, we notice that they are not equally distributed in all organisms. In the red lineage, we mainly find LhcX, while in the green lineage we mainly find LhcSR and PSBS together. However, in vascular plants have lost LhcSR and have only kept PSBS for photoprotection. The presence of LhcX in chloranthionophytes is likely the result of the secondary endosymbiosis of a green algae chloroplast into a red algae, which has already LhcX incorporated. The different distribution of LhcSR and LhcX, as well as PSBS in algae and plants, is not surprising if one thinks about the different habitats, like the oceans, the flat coastal waters, or land, which they inhabit, and the different light intensities and light environments that they face. So they needed specialized mechanisms to deal with these circumstances, and to have photoprotection. In the end, all of these topics are very much still subject of interest of research in the photosynthetic community in particular, and not all details of light harvesting antennas and the molecular massing of photoprotection is known and are still under debate. However, I hope that this lecture gave you an overview about the evolution of photosynthetic eukaryotes and some details about the differences in the light harvesting antenna proteins. Thank you for watching.